Hello, my name is Tara Choudhury, and I'm a sophomore at the Academy for Allied Health Sciences in New Jersey. My mentor was Alina Patel, and I did my research paper on single-cell gene expression of the tumor microenvironment of brain cancer. My research paper is a literature review and a computational analysis of the full set of genes that are involved and asks whether the cells of a tumor's environment are completely like the cell types they are described or if they express some abnormal genes. So I picked this topic because I like the idea of doing my project on cancer because I was always thought it was interesting of the anatomy and physiology of how a single cell could clone itself into a tumor, and also the fact that cancer can live through almost anything as it does not react to some treatments. And I also want to go into the biomedical engineering field, and single cell RNA sequencing showed me one of the many things you could do in a lab. So I wanted to start off with what is cancer? So cancer is a disease when an organ has a gene or multiple sets of genes that mutate so that cells uncontrollably divide and interfere with the functioning of healthy tissues and organs. Cancerous cells can originate from specific sites of the body or multiple different organs. When a cancer is metastatic, malignant cells can travel to parts of the body and invade healthy organs. These cancers travel through the bloodstream and can invade places such as the brain. So next, what is single-cell RNA sequencing? The purpose of single-cell RNA sequencing is to extract genetic information from tissue cells and label them for analysis. Single-cell RNA sequencing works by separating single cells that originate from tissue samples, example being tumorous tissues, into a tube. Then the cells go through a microfluidic device that takes in the single cells and suspends them into nanodroplets. Then the genes are barcoded and amplified by PCR. The genes are then discovered and put into a computational library where they can be looked at online from anywhere. They are then analyzed and plotted in diagrams such as the TSNE plot. Single-cell RNA sequencing reveals the composition of different cell marker genes, which identifies the different types of cells and their functions in an organism. So how I got my data was downloading RStudio for Windows onto my computer. Then I had to download Seurat and Seurat object packages. I downloaded the genes from the Gene Expression Omnibus, and I uploaded the various files into RStudio. This picture is a piece of my code that I used to generate a TSNE plot using Seurat and RStudio. This code plotted a single-cell RNA sequencing data set with labels depending on the genes found in the different cells. So tumor samples that I used came from 5 patients that had metastatic tumors in the brain that originated from either the breast or the lung. This picture shows the different cell types clustered under labels of what cell they are. This is dependent on the genes they express. Every single dot on this graph represents a single cell. I found it interesting how there was a big unknown region on the graph and that it had a small amount of tumor cells in it. However, there are no cell types that specifically claim to be a part of this unknown region. The majority of the cells had tumor genes. Next, I would like to go into the different types of cells that I found. One of them being myeloid cells, which are a white blood cell that plays a crucial role in the immune system and the body's defense against infections. Myeloid cells can stimulate an anti-tumor immune response, but more often they stimulate tumor development, including promoting invasion, tumor growth, angiogenesis, and metastasis. Next, the oligocells, or the oligodendrocytes, are attached to myelin and wrapped tightly around the axons. Finally, the NSC cells are neural stem cells that are most commonly apparent in the development of a baby. However, sometimes they are found in adults. This is another graph, the same as the last, except using a different data set from the 5 that I captured. I observed significant heterogeneity of the tumor cells in terms of their gene expression and analyzed by single-cell RNA sequencing. Heterogeneity means a full spectrum or consisting of diverse elements. In all the data sets I looked at, the fibroblasts and the endothelial cells appear to be very similar in areas, as the computer couldn't tell which was which. The fibroblasts are the most common types of cells in connective tissue and are present in many parts of the body. Endothelial cells form the endothelium, which is a thin layer in the interior surface of a blood vessel throughout the body. I even observed some tumor cells that appeared to be similar to T cells, which was overall very different from the majority of the population of tumor cells. This being that T cells are a type of white blood cell called lymphocytes, and they help the immune system fight germs and protect one from disease. Moreover, the unknown regions, as mentioned prior, had some tumor cells in them, so it's unclear if there is an unidentifiable gene that codes for a tumor cell that the Seurat package did not know of. I go back to my initial question when I was asking whether the cells in a tumor's environment are completely like the cell types they are described as, or if they express some abnormal genes. Because they were metastatic tumors traveling throughout the bloodstream, there were different types of cells that traveled with this tumor to the brain that changed the tumor microenvironment. So not only were there nerve cells, but there were also tissue cells, blood vessel cells, immune cells, etc. Also, while they may resemble the cell type they are described, they frequently express abnormal genes due to mutations, epigenetic changes like behavioral changes, or interactions with the surrounding environment. Thank you.
Wonderful, Tara. Thank you so much. We're going to open it up for questions. Let's see if questions come in. First question for you. Do you think AI will be able to help analysts understand computation better in the future?
I do believe AI will be able to, considering they can go through all the steps that I had to go through probably much quicker because I had to form different other graphs in order to piece together the information to make the TSNE plot. So an AI could do so much quicker and get the results much more quickly.
Awesome. The other question is, could this research lead to new treatments that target these specific cancer cells?
Yes, I do believe so, because once you go break down the tissue and you go to the individual cells, you can see how they interact with each other and what are expressing what genes. So then you can see, okay, this is experiencing this certain gene, so I should target that to turn it off so that there's no more tumor like uncontrolled regulations.
Awesome. And the last one is, what drew you to this research topic?
Um, when I was younger, I was watching the TV about all these different research discoveries, and I always liked learning more about them. And then I was always drawn to the anatomy and physiology of just cells in general. And then I got drawn to cancer, and then I learned about single cell RNA sequencing. I decided to do my topic on that.
It's fantastic. Wonderful. Thank you so much for sharing, Tara. That was fantastic.